Testing 1 2, testing 1 2. Testing 1 2, testing. Okay, I think the music needs to be a little louder. Come on, cursor, behave. Let's go. And sometimes the cursor does not want to behave. There it goes. Okay, good stuff. Hey Teeny, hey Gary, how's it going? Another impromptu thing. I wanted to go through some water samples one more time before I get rid of them. Back to nature. And I don't know, it's kind of an experiment, I guess, on how long you can keep them in the fridge and still be viable. I do open them up periodically to air them out, but I was just curious. Also, I have some other random crap from around the kitchen and some other plant samples I didn't look at before. So, first things first, I thought I would show. I also changed the layout a little bit, just experimenting with OBS. So let me know if this works out okay. But I have a bigger view area now, so it's actually got a little bit of a blur there. Let me fix that. That should be so slow. Let me fix that. Of course, when you change the exposure, it also messes up the bit depth. You also change the exposure, so I think that should look better now. That should track a little smoother now. Yep, nice and smooth. So I added a little desk area now so you can kind of see where I'm working. It's not optimized yet, but these are the 4 vials I'm working with. Got some stuff from the kitchen to look at. There's a side view of my dissecting scope here, also called a stereo microscope. But I'm starting with a compound as usual. So give me a holler, let me know if you can hear me okay and the sound balance is good. Rearrange my images a little bit here. No, don't put that in there. OBS is getting weird here. Okay. Yeah, I thought I had spent some time with OBS to get the layout a little bit better. Just that was my bad. I hadn't had time to tinker with it much. It was just more like get the information out there, start streaming. But today I messed around with OBS a bunch to get a slightly better layout. I had to make this camera quite small, but anyway. So what we're looking at just for fun, because I was eating some earlier, is this is what Nutella hazelnut chocolate spread looks like under a microscope. So you look at it with your eye and you say, oh, it's solid brown. And just like if you ever looked at blue jeans or something other similar color that's fairly blue, maybe not solid blue, but pretty blue, but your eye perceives it as overwhelmingly blue when it's just some blue fibers going through some white fabric, basically. So it's just your eye fills in the blanks, makes things look more brown in there. Here you just see little flecks of, in fact this is probably a little bit less brown than this. This is actually a little more, what looks like a little more bleached out. So most of what you're seeing, the flecks, to give it the brown, the chocolate flecks, are those kind of reddish, orangey bits. And collectively, those add up to be the kind of brown. See, it's a thicker portion up here, so it's getting more discolored. But all these little specks of brown here, that's the actual chocolate specks inside the fatty matrix that makes up the chocolate, as well as little tiny bits of hazelnut flavor and all that kind of stuff. So this will be kind of fun. I was eating some earlier. I go, I wonder what this looks like under the scope. So this is the compound view. Just something for fun to look at. So, but again, just to give you the idea of your perception versus reality. At a macro level, you're seeing solid brown and smooth, and of course at a micro level, you're getting more individual little components that are representing all the different colors making up the brown, as well as the kind of matrix that it's in, that's just fairly nondescript and just kind of well, it's fatty basically is what it is. And there are probably some sugar crystals in here that aren't fully yeah, it's not very it's pretty smooth normally, but I suppose if you give it some different temperature treatment, you might get some crystallization of the sugar out of the spread. So just a quick fun thing, and I will look at it a little bit later in the lower power, just to show what that looks like. And just for fun, I also did some, I haven't looked at calcium carbonate for a long time under the microscope, and this is a smashed Tums basically. Not super smashed, but also not very spread out. Let me, there's way too much dark blocking the light here. Let me fix that. Make it a little more sparse. Not enough light getting through there, just show the calcium carbonate texture. I didn't totally powder this from the Tums, it's still a little bit chunky. So it's not like the calcium carbonate powder you'd get from a lab. So that's actually still pretty heavy. It's not letting a lot of light pass through it. Let's go back out a level and change the exposure a little bit. So you can see the color added to it because it's a pink Tums. And let's see. Maybe zoom in on that part a little bit. And change exposure. So you can see the granules a little bit. It's not an awesome thing to look at, unfortunately. I just thought it'd be kind of fun to, if I could powder enough to get a texture out of it, but it's still kind of blocky. It's not wonderful. But you can see the color. This is pretty uniformly colored, unlike the hazelnut spread the Nutella was. Because this is a dye added to the thing. So even the fine bits have a little bit of a tint. It's not like it's white and then you need a whole bunch of it to get the color. Obviously the big chunks here, let me know if you can see, you guys can see that cursor, right? I think you can. Move this over a tad and see if you guys can see. The cursor should be showing up. Not seeing the cursor on the field. Oh, no, there it is. Okay, it's showing up. Okay, so that is just a quick look at some random junk that I had sitting around. Not wonderful, but it was just something to look at real quick. Let's go on to some water samples. And again, I'll look at those again under different power on the stereoscope. So let's go back through these samples again real quick. This is sample 1, which is probably still not great because I have memory serves. It was probably the worst of the water samples I had. Let's get to sample here. This has got a bit of a tint to it. It should be more neutral. Let me adjust this a bit. The camera's got a bit of a tint to it this time. I think when I messed with the color balance here, there we go, it's a little more neutral like it should be. Okay. Much better. Alright, let's fix the bring up my adjustment window again so I can get a better focus. Sometimes I get a slightly different focus between the scope and the camera represented on the stream, so it's easier if I do most of the stuff driving with the screen, although the image is actually better quality in the scope itself. So let's see, I've got a lot of the usual plant fragments in here. More junk. Take a few diatoms there, a little cluster of stuff. Its exposure's way off here, hold on. I think we got a problem with the camera setting again, let's fix that. It's better, and I'll fix the rest with the scope. Zoom in a tad. Get some diatoms here and some other algae. Gotta tweak this stupid exposure again. Better there. Okay. Alright. So yeah, we've got a few diatoms sticking out at different angles there. Some more green algae I'm not familiar with, kind of clustered in the middle there. So it's something. Not seeing a lot in this sample. So cyanobacterial threads poking out of some debris there. More random junk here. Let's go back out to lower power. There's a lot of big stuff in here. Ah, this is another piece of that large tubular algae I had the other day. Totally forgot the name of it. I put it on the stream title of the last stream, but it makes this really cool tubular lattice work with these alternating hexagons and pentagon structures of rings of cells. This is a fragment of one of those, and the name is slippy meter now, unfortunately. Let me fix the lighting. And let's try a quick digital zoom here just to get a quick boost. And you can't quite make it out here in this sample because it's kind of smushed. But the footage I had earlier on a previous live stream, this is a beautiful lattice work, all stretched out. And you can see the structure really well. This system's kind of smushed together, so you're not quite seeing it the way I'd like to see it. Let me go back to look for something else here. That's a lot more than I found the last couple times I looked at this sample. Oh, there we go. We've got, is that a rotifer? Very nice. Let's try and zoom in here a little bit, try and get a better look. Yeah, it's a fat little rotifer. Fix the exposure. It's a chubby one, looks like a little ghost. And he's moving a lot. This is going to be tricky to trace it down here. Fix the lighting a little bit here. Man, this guy's fast. Beautiful though. It's usually a little fatter than I'm used to seeing. They're usually not this wide. I'm having to constantly focus and cruise around different directions. Sorry, I haven't had a chance to look at the chat yet. Now I've got a rotifer keeping me busy. Going up and down in the Z a lot. I'm constantly focusing to try and keep it in frame and focusing in the center of the frame. It's got some nice refractory effects though from this little rainbow. The optics are working out. Ooh, he's going. Go, rotifer, go. Finally took a break. Here we go. Checking out that air bubble. It's finding some food there. It's taking a little break and then it's going to take off again. Come on, buddy. Whoa, out of focus again. It's up and down way too much. This is tough. I probably put a little too much extra water on the slide, so I give him a little more leeway. Nice tumbling, that's cool. Go on, tumble. It's an active little bugger. Chase him a little bit more, but you guys might start to get seasick here a little bit if I keep moving too much, so I'm going to try and let him come into frame a little bit. Also got because I did a digital zoom, I've kind of got a little bit of a mismatch with what's in the eyepiece versus what's on the camera. Try to get it more centered. Okay, so he's not. Keep him toward the top of the frame, then he's more centered on the camera. It's fun to watch it move around. It's tumbling a lot, twisting, and it's an active little guy. Probably had a little too much. That might help a little bit with the contrast. Now it's doing loop-de-loops. Yeah, it's just moving so much, it's really hard to stay in focus. Nice specimen, though. Fix the contrast a little bit more there. Camera's not quite doing it justice. The microscope looks like a little jewel. It's just sparkling and really cool watching it tumbling around. Yeah, it took a break now. I can stay focused on it for a tad. It takes off again. Let's fix this. Better exposure on this guy. Yeah, it's catching the light just right. It's kind of funny. It's got the diffraction gradient effect on the outside. A rainbow action. Take a chance now to center this a little bit better based on what the camera's doing. That's pretty good. Just having fun probing around this gunk here. Pretty good though. Slide 1 was boring last few times I took it out. This batch, motor batch number 1, so I'm happy to find this. A second to catch up on chat since this guy's taking a break here. Oh, hey Manny, sorry I missed you earlier. Just doing a mad chase with rotifers here. Yeah, I'll get the sample 14. That's the good one. I don't want to start with the best one and then have the letdown later with the not so good one. Oh, what's going on with this guy here? Fix that. Oh, why did that do that? I didn't mean to do that. Digital zoom crap. Okay, that's better. That is better. Probably push a little bit more without having to change objectives. So, problem if I go to the next objective, this is 100 power plus a little digital zoom. If I go to 400 power, it's going to be harder to focus. Also, that's my wonky objective I need to replace. This should look pretty decent if I dial this in. It's only a bit more. Don't make it too dark. But yeah, this guy's pretty interesting as it's tumbling around. light glinting off of it, different angles. Let's check the composition here. It's not too bad. This camera's not dead, right? Okay, it's live, good. This happened earlier. The camera software, probably a stupid Windows thing. Camera software is now stuck. I can no longer move the window around, so I'm going to be stuck with it the way it is right now. And I probably can't change the digital zoom now either. Just going to make it interesting. No, I can. So why is the window stuck? Yeah, sometimes Windows is really annoying. Last couple of streams I did were on the Mac because I didn't have a Windows machine reset up. So I had to reconfigure this one to do the stream, and as usual there's some technical issues with it, although it is working mostly. So yeah, this rotifer's having fun. Let's go on a little bit more stuff on this slide. Let me back out. Fix the lighting. This is kind of where I need that stupid exposure control on the Windows software. Now it's letting me do it. I must have a little timeout or something. Adjust it from here, and then it's not so blown out. It's just a focus issue. Okay, there we go. Cool. What else we got here? Yeah, that's actually pretty much it for the slide anyway. I was at the edge. Alright, that was number 2. Let's go to number... Oh, sorry, number 1. Let's go to number 2. Nope. That's not number 2. And that's the good one. I'm going to do number 2 next. So, 1 was the little waterfall near one of the fish tanks at the zoo. This is the tank, the water from the actual cichlid tank. There's a bunch of African cichlids, little fishies from Africa that are usually quite predatory in nature. And you have to breed certain ones together, other ones fight and try to kill each other. So, you have to be careful with them. That's what we got in this one. See what we got in this one. I used so many slides last time, I had to clean a whole new batch of slides. Alright, let's go all the way over. So low power with a little bit of digital zoom. See what we can see. Lots of debris as usual. Air bubbles there. That's nothing. Looks like it could be something from here, but it's not doing anything, so I'll bypass that altogether. Lots of other air bubbles. Let me fix the exposure. That's a little too dark. There we go. It's like a star field. Let's see. Junk, junk, junk. I'm going to skip number 2. Next time I go there, I'm not going to get a number 2 sample. That site kind of sucks, which is a good sign. It means the water is very clean there. The fishies have a nice, happy environment. No good or bad protists in there. So that's actually a good thing. But not for this purpose. I want to see things. Let's try number 3 next. This is from the pond that feeds the filter trap of sample number 4, which is a really good sample. So there are some things in here, but most of the goodies end up in the filter trap. So far anyway, we'll see what happens now. Fix this here. So yeah, not much to see on the camera here with the my secondary camera with the table here, but when I get to the macro stuff and the other scope, it'll come into play a little bit better, I think. Just so you can kind of see what I'm working with. And just to have a separate camera angle just makes it a little more dynamic to look at, especially on the playback when people can't chat or whatever. Okay, what do we got here? Let's go to this side again. Get this focus. You can see that cover slip is kind of sliding around. I put too much water in the slide. So when you do this, cover slip kind of jostles on top of the film of water, like it's floating on it. Good old surface tension. More debris. It's just a constant plant fibers everywhere. Which makes sense. I mean, it's made of cellulose and other fibers, so cellulose is incredibly durable. Only certain organisms tend to eat it. And in some cases, like giant redwoods and even coast redwoods, their cellulose matrix that makes up the trees, along with the terpenes and other compounds inside of it, make it also very unpalatable to the critters that normally eat wood. So you'll find old sawdust piles from sawmills up in the Sierra Nevada mountains that are really, really old because nothing broke the sawdust down, or it's just taken a hell of a long time for stuff to leach out of the sawdust for it to be palatable for critters. So that's why redwood's such a good building material for decks and stuff outdoors, because it's super durable. Nothing really wants to eat it. And that's without chemical treatment. Normally you have to use those like copper naphthenate green pressure treated boards with the big holes in them. They have a greenish tint to them. Those are really nasty, toxic things. You don't want to touch them with your hands if you can't help it. And because they're treating nasty stuff, nothing wants to eat it either. Okay, so again, nothing but fibers and air bubbles and stuff. It's kind of boring. Makes some interesting bio art for graphic design, but nothing to look at here. So sample 3 is a wash. And you know what that means? It's time for sample 4, which has all the goodies. Give it a nice little shake so you get it all stirred up in there. Okay. What have we got on this one? Already got some little protists moving around, some little flagellates, tiny little things dancing around, little particles. hard to see. I can beef up the exposure a bit so you can see it's like a little star field. And you'll see the parts that are moving around. Little tiny protists, little flagellates and stuff bouncing around, but too small to zoom in on and get a good view of or follow. It's pretty fast. There's some cluster debris there with the little cyanobacteria filaments in there. Makes it look hairy. But nothing grabs me yet. Those are possible little islands. Remember I was talking about other shows, islands of activity, like little forests or like little coral reefs, micro coral reefs where things are going on all the time. And it tends to attract things, so it's fun to just explore them, hang out, see what's going in and out of them. Little magnets of activity. So far those aren't so great. Let's try this one, there might be something here. Fix the exposure there a little bit. Let's go in for a closer look. See who's here. Okay. There are some little things bopping around in there. You can see little hairy things sticking out of the top. Fix the exposures, you can see those a little bit better. Might look better in this view. Bright field view. And I change the exposure. There's those little hairy extensions going up and down. And little things cruising around there. And let's go to... Yeah, they're coming in and out of this stuff, but they're very, very small. Not for anything really big to look at that's cool. Yet. Stuff, but it's not that exciting. Let's go to the one over here. So this is what happens if you have too much water in the, so this one's outside the cover slip, which means it's even harder to focus through because it's not compressed. The depth of field is way too much. So I need to find stuff inside the cover slip. There's a little guy cruising around there, kind of in the washed out area. Might get a closer look at that just for kicks, but way too much exposure there. There it is. Tumbling, tumbling, tumbling. Clearly from the emotion, a flagellate. It's very wiggly and kind of going all different directions. It's not a smooth swimmer. Lots of tumbling. And then it'll start to go super wiggle with the flagellum and then it starts going all different directions. But it's kind of fun how it does a nice little tumble. To stay there and focus, you can see the tumbling. A bit more light on the guy. Tumble, tumble, tumble. Really nice tumbling motion. It's going up and down a lot, so I'm having to focus constantly here. So yeah, it just has this kind of gentle tumbling, and then all of a sudden it goes, ah, and just wiggles and goes in a certain direction. with more purpose. And then it goes back to tumbling again. This is not centered quite right. It's kind of heading toward this debris here at the top. Oh, there's another one right here inside the debris. Crawling through, or swimming through. But they get lost inside this stuff and you can't really track them down. There it is at the bottom. Yeah, it's behind. I have to focus behind the stuff to see it. So there's still plenty of life in these samples. I'm not finding any really good big stuff yet. But there is still stuff here. So that was a kind of a boring slide. Let's try another one. Another sample 4 slide. This time I'm not going to stir it up as much. See, stuff kind of hovered to the top, floated to the top. Let's see what we can see. This was a risky stream just because, again, I'm not using fresh samples. So you never know what you're going to find. Last 2 episodes were very, very good for this stuff, but at this point it's getting to be old samples, so I wouldn't be surprised if I didn't find a lot of really cool big stuff in it. Lots of little things that don't seem to have trouble living off the stuff that's left over. But not finding a lot of stuff here. A few small things bopping around in there. There's always something in this one of these slides, in this sample, because there's some various size. A little diatom there. Looks like that might have been a ciliate that's dead, but it could be something else. This looks like a piece of debris that's just the right shape, but it has the look of a couple different ciliates have that kind of look to them, but it's definitely not moving, and there's not enough structure there to really look at. So bypass that. More filaments of junk. What's this? Air bubbles. Much going on here. Air bubbles, diatom. There's a couple little flagellates bopping around here. I'm getting too much water. It's kind of sloshy when I bump the table by accident. Oh, what do we got there? That's interesting. Got some structure, but it looks like a pile of things. There's some blue-green algae threads in there. It's too oddly distorted to be an air bubble. But it's also not doing anything, so I'm not sure what that is. Very interesting shape there. More fibers of junk. More fibers, nothing moving. Nothing. Get up here. A diatom there. No island of stuff. But nothing moving except for a little flagellate next to it. Nothing here. Yeah, so this sample may not be as good as it was. Kind of expected that, but let's make one more attempt, maybe two, and give it another shot. This time I'm going to grab some of the darker debris and let it settle a little bit. This is where you get a lot of the, found some of those worms. There's a polychaete worm and another type of worm which I haven't identified yet. And nematode, and there's a copepod in there last time. There's all kinds of stuff in there. Color in it. I picked up some more chunks to look at. Okay, lots more chunks. Definitely more flagellates bopping around. Of cellular debris of this and that. More little islands with filaments and green stuff. A little tiny island there. There. Okay, nothing there. There's another little diatom colony thingy with some filaments and diatom backbone and some other junk collected around it. It's like when you get like big chunks of kelp and other seaweed on the beach and other things attached to it. Except for some little flagellates. Nothing serious here. More little clusters of junk. Big old fibers there. Oh, a diatom shell there. Nice pointy one. And some other bits and pieces of stuff. But nothing really to watch, nothing moving. there. So let's go up from here. Another little cluster of stuff. Little hair sticking out, little filaments. There's another little flagellate bopping around that island there. Another island here. Nothing happening around that island. Oh, there's a bigger one. And one next to it, but again, nothing too exciting. Lots of tiny stuff, but not finding any good big stuff. Most of the big stuff has died. Lots of little things. One little guy swimming around here. Another chunk of stuff. And a chunk next to it. Oh, big old chunk here. Yeah, this is part of that shrimp that they feed the flamingos. That has that looks orangey on here, but it's pink. This is part of that optical illusion where, like I was showing you with the Nutella, looks nice and brown far away, but up close, it's more of this is like a pale orange at this scale. Oh, there's a guy cruising around the debris. That's something to look at. And a guy swimming around the outside of it. Again, the same kind of small little flagellates from before. So, not a lot of big stuff here. This piece has some depth to it. You can see when I focus through it, it's quite thick. And there's some things swimming about this cluster. Cluster there. Yeah, nothing big in here. At least not anymore, or I'm just grabbing the wrong section of the vial. I'm going to try, now that everything is settled, after I stir it up a bit, I'll try one more slide from the juicy bits on the bottom. It's quite dark on the bottom, so I'm just going to get a nice section right out of that. That's pretty dark. Let's see how teeming this thing is. Teeming with critters, I hope, or at least what's left of critters. Oh yeah, I'm already getting a lot more stuff in here compared to the last trial I did. But again, mostly little things. I'm not getting a lot of big stuff in here. Oh, I spoke too soon. There's a worm. That's what I should have done the first time. Just go right to the bottom without mixing it up. Zoom in a bit. And got another one of those cool little worm dudes here. Or actually, could that be... No, it's not a worm. It's one of those long ciliates, like... oh damn, the name is escaping me. I said it on the stream that I had with the worms. There was a couple of these guys in there along with nice. Look at that. Spirostomum, I think it is. Anyway, it's real long and flexible. Look how flexible it is. It's going in and out of that thing. Let me change the lighting a bit. So it's a little more easy to see. There's more of natural lighting, the bright field. So you can see the color of it a little bit. We'll go back to the dark field so you get some nice contrast and color. See, I think this is Spirostomum, but I have to look this guy up. Real long. You can see the cilia on the sides beating as it's, but look how damn flexible it is. It's one of the most flexible ones I've ever seen. Very worm-like, but it's one giant cell. Whoa, what was that going in there? Something to zip through there. This is not quite centered. Let me fix this. Now it's pretty centered, I think. Oops. Zoom in a tad at least. Get a little more power out of that. It doesn't like it when you do that. There we go. Get a hand out of there. So again, loves the debris field. Just going through. This is like little pieces of shrimp parts and other fibers. There's some diatoms in here. That's a nice one. I'll come back to this one. Spend a little more time on it, but I want to see what else is here. I'm dying to see. Since this one's so much better than the last cluster. More filaments there. This guy. Fix the lighting a bit so you can see the filaments. Another section of material with lots of debris but no big guys. Fix exposure here. Nice stacked cell structure there. I think this is also part of the shrimp. That the flamingo is like a more bleached out part. It's not so pink or orangey compared to that piece, which is much darker. Diatom cluster. Remember, this came out of a filter trap, so they were probably sitting in the filter for a little while and stuff just kind of got smushed together and just stuck. So you get these little colonies of things that form from water flowing through it, just kind of getting stuck together and just attracting each other after a while. Ooh, nice piece of shrimp antenna there. Look at that. Big, hairy bit sticking off a stalk. Of course, more diatoms. We got some other algae filaments going through here. Some shrimp pieces. Some nice algae filament there. Nice green guy there. Quite long. Looking for movement here. For some action. So not much going on there. Back to this dude again. So this is a nice place to come back to. I'll remember this one. I'll come back over here. Let's head over to another area. that's interesting. Look at that. It's like something that split open. And with the amount of different food and stuff in the pond and plant seeds going in there and all kinds of stuff, this could be anything, but it is pretty cool looking. Just gonna show it, not speculate on this guy. More junk, more junk, more junk. Nice, nice algae thread there. Very cool. Kinds of colors and things going on here. Oh, there's another. Oh yeah, it's one of those guys, Amphileptus or something like that, poking around through the debris. It's got a long kind of a bowling pin shape to it, a little more taper than that, and the top is also very flexible, like a proboscis. It's again one big cell, but the way it moves around is very much more complicated than something like a paramecium. So I keep finding these guys, which is really nice. Could probably, I could probably push it one more objective. Well, this is going to be a focus nightmare. Oh, there it is at the bottom. So it's in and out of debris, it keeps folding on itself. There's that floppy front end that acts like a proboscis. But I'm looking through a bunch of debris too, which makes it unpleasant at that. Come back out. This looks a little better, even though it's smaller. Get a better idea what it's doing here. And I could probably push the digital zoom a tad more. The center. It's actually in the debris. You have to watch it peek out. You can see it kind of flopping around a bit. Catch up on chat here. I'm way behind on chat. Sorry, guys. Got too busy looking for stuff. Sorry, I missed Teeny. Teeny left. Hey, Henrik, how's it going? You don't understand? Yeah, sorry. I'm throwing out names of things and some terminology here and there. Just it's an educational stream, so I tried to make it too nerdy and too technical. Of course, a lot of the stuff I also forgot over the years too. I need to spend more time reading recently on some of these things. Some of these I haven't actually identified because they can be difficult to get beyond a certain basic level you know, the generic term of flagellates for something with flagella swimming around wiggling, and something with cilia being smooth and gliding around and tumbling. It's a pretty basic term, but for these little guys that are really hard to get close to and follow because they're so fast and they're so tiny, you pretty much just have to identify them by their movement and just kind of put them in a rough category, which is, you know, good enough for most people. There, not too crazy about it, but some of these things that have a lot of structure to them, even though they're a single cell, like this guy I'm looking at here, which I think is Amphileptus. They're just incredibly flexible. You look at something like a paramecium, it's kind of boxy and can flex a little bit, but it's kind of stiff. And then you look at something like this guy, which is flopping all over the place, or like a blepharisma, or the other one I showed earlier, the spirostomum. They're just amazing. Long, flexible cells that, because of their flexibility, act more like something multicellular, like a small worm, or such. So it's really a trip. Yeah, I would like to go closer, but that stupid objective's bothering me. So, well, that guy's flipping out in the space there. Whoa. So I found this guy again. I like those guys. Let's keep going. I'll go back to the spirostomum thingy later on. It's pretty fun. Just looking for big stuff. Oh, got the end of the cover slip, so just one quick scan up. And go back up to hang out with the nice wormy looking ciliate. Yeah, nothing here. Let's go back over here. This guy's hanging out. Has a nice piece of shrimp bits. It's down here somewhere. Should be in here. Unless it moved. Oh, there it is. Yeah, look at that guy. Nice, long, stretched out. So at this point it looks like, oh, okay, it's just a long, thin ciliate, right? But then watch this behavior. It's long and incredibly flexible. Like it's more like a worm. And it's really fun because as it twists and turns and flops around, you can see it even tumbling on the longitudinal axis there. Tons of, it's just covered in cilia. You can really see them well. It's like a halo around the body. Makes it look kind of fuzzy. And they are just incredibly huge and flexible. This is only 100 power plus a little bit of digital zoom. So they're very large. Paramecium at this scale would be probably 75% the length of this guy, something like that. So it's a big guy. And you can see all kinds of stuff going on inside the cell. Not only is the cilia bouncing around, but if you happen to get a clear shot of the body as it's tumbling and stuff, there's so much stuff, activity in the cell, it looks like it's sparkling. Really interesting. This one I can push a little bit, I think. Let's try a more objective push. Get a nice close-up of the body there. A little more light. Change the contrast a bit. So you can see, I have to focus through the body a little bit, but you can see the cilia beating on the margins there. Oh, did it just leave? I think it just left. Yep, it just left. Let's go find it again. Didn't like that spot anymore. It's not there. And it's looking for more stuff to check out, look, apparently. I never, oh, there we go. It's going between the little islands. Okay, make up your mind. Can't quite decide. It's on a little pattern. Alright. Just going to bend around that piece. See, very flexible. Like you would see a little aquatic worm. Look at that, how it bends and flops around and stuff. It's pretty impressive for a single cell. It's crazy. I mean, the ultimate example is like an amoeba, but the amoeba is such a strange. It's like how our white blood cells work. It does this kind of phase transition change of the cell structure, where it becomes more liquidy in some parts and more solid in other parts, and then it puts out those little pseudopods to spread out and grab things and surround things. But it's not so much flexible as this is more liquidy. This is still quite, keeping its shape and rigid enough, but also incredibly flexible, whereas a paramecium is kind of boxy. So they're just amazing. Just waving around back and forth. If you watch something like a nematode, which is a multicellular worm, not actually about the same size as this guy, which is pretty crazy, because multicellular, like 1,000 cells versus one giant cell, yet it's about the same size. It's totally wacky, the stuff at this scale. And you watch the behavior, and this is very similar behavior to a nematode, but nematode, because they have these longitudinal muscles that are contracting, making it move very whip-like, super quickly back and forth. It's more of a frantic thing. This has more purposeful moves, a lot more gentle and stuff, whereas nematode just appears to be just like thrashing about. So it's supposed to be a more advanced animal, yet this one looks more, for lack of a better term, calm and civilized than a crazy nematode flopping around. They're just fun to watch. Very fun to watch. I'm going to adjust it so you can see that the back part of the body gets kind of lost if I don't change the exposure just right. Because there's kind of like nothing there. It's just a void. It's cell membrane with a void in there. You can't really see it. It's like a dark space. And add a little bit of oblique lighting to it. Looks a little more 3D. Oh, you see that contract there. Didn't like that. It got a little small. It's a defensive move. There's going to little crevices and because these have a specific kind of, again, lack of a better term, body plan, it's just a cell. But they're differentiated enough to be more like a head and a tail. So you can see that part that keeps going in and out is like the so-called head region, and this little dark area at the base is the tail region, because they always lead with that front part there, which is closer to where the kind of oral groove that they have is located. The paramecium have a similar thing. It's like a kind of a furrow in the cell membrane. It acts like kind of a primitive oral cavity, and they funnel the bits of bacteria and other stuff that they're trying to feed on. The really small stuff. The small flagellates you see bouncing around, they're actually too big for a lot of these things to eat. They will occasionally catch them, but they have kind of this rejection filter, which is the oral groove, and then they have at the kind of base of the oral groove inside is where they can envelop particles with the cell membrane and form vesicles, and the vesicles bud off and go inside the cell. It's like, folds in on itself and buds off inside the cell. So it's a very simple packaging mechanism to make individual little vesicles, little food storage things, kind of like micro little stomachs that kind of go inside the cell and get funneled to a location to kind of hang out, get digested, and then from there the contents can be transferred to something else as the vesicle degrades. And it depends on the cell you're looking at. These are all eukaryotic cells I'm looking at here, which is they have a true nucleus, much more complex organelles and stuff, structure compared to something like a bacterium, which has no nucleus, or also a blue-green algae or cyanobacteria, which I also talk about, those little threads. Those are more related to bacteria than they are to these other animals and plants, because they don't have a nucleus. They don't have organelles per se to do all the processing. Like they don't have mitochondrion, they don't have chloroplast, they don't have Golgi apparatus, endoplasmic reticulum, all the good stuff that's part of a standard eukaryotic cell that everybody studies in basic biology class, which basically makes the factory of the cell. The cell basically, just like any decent company that has shipping and receiving, import-export, engineering, manufacturing, packaging, all that kind of stuff. The cell does all that stuff. The complex cell does, the eukaryotic cell does that. Bacteria are more kind of loose. They have like a ring of DNA floating around the cell called a plasmid. They don't have a nucleus. They don't have organelles. This is the whole concept of they've changed the name of it recently, or at least a few years ago when I stopped kind of keeping up with the stuff. But there was a thing called the endosymbiont theory, which is basically the concept that a more primitive cell captured another cell, possibly during a feeding of some kind, or some kind of merging thing, and it basically that cell became the differentiated organelle inside of it. So a mitochondrion at one point was some other prokaryote that was captured by another cell, and because it didn't digest it, it didn't die, it started working symbiotically with the organism. And mitochondria have their own DNA, so that's why they another reason why they think that is the case is that no other organelle has its own DNA. Basically, mitochondria and the chloroplast do as well. That's more evidence leading toward the concept that at some point it was a captured prokaryote that became part of the cell, and because it didn't just generate it, it basically had to capture it and then make a symbiosis with it. Just like you have on a larger scale, you have mycorrhizae, the little fungus nodules that live off of roots of trees in certain forests, and the roots and the mycorrhizae have a symbiotic relationship. They provide water and food for each other back and forth, and protection, and they cooperate. Or like a lichen. A lichen is a fungus body with algae living inside of it that gives it that color, like a green color or reddish color or yellow. And that's the algae inside is giving the color and providing the photosynthesis for the group, and the fungus provides the home. So on a smaller scale, that supposedly happened with these individual cells, and eventually our cells. Yeah, it's fun to watch. I could just watch this guy all day. You see when it rotates, it has a kind of thin ribbon shape to it. It's actually twisting, especially on the back end. You see that kind of ribbon twist because it's flat. It's really cool. Just change this lighting a bit so you can see a little bit better. There you go. So there's that ribbon twist in the back. Just hanging out, looking for yum-yums. Again, nothing to see at the so-called head end, but that's what it leads with, and you know, there must be some slightly more sensory bits toward that end, so it can know to go. That's always the gullet, you know, like the oral groove side. So they don't have eye spots like something like Euglena has, which actually has chloroplasts in it. It's actually an animal with chlorophyll, which is a trip. I've caught them on the stream before. They're pretty cool. That's when it gets really confusing, because you go, what's really an animal then? Why does an animal have chlorophyll when animals are basically what you call heterotrophs, meaning they live off the environment, they need to take in nutrients, whereas plants and algae and stuff like that are considered autotrophs, meaning they self-feed, they make their own food from chemical, you know, sunlight converting chemicals to stored energy. So but here's an animal that can feed or make its own food, or both. It's kind of a freak out when you think about it. So that's this guy. And again, I think it's Spirostomum, but I have to look it up. I forgot. There are only a few ciliates that kind of have this long body plan and very large. And also this behavior is very, very flexible. Just fun to watch. Look carefully at the front. There's a little bit of like a fuzziness. That's the cilia going crazy. And you're seeing it at the length of the body, but it's really pronounced at the front end at this light level. Oh, and there's a little guy next to it. Is one of the Amphileptus guys. Look how much that thing stretches out. Holy smokes. Again, one single cell acting like a much more complex organism with that flexibility. So two cool guys in one frame. That is really neat. So that, look how much flexibility that thing has. It's flowing like water at times. More fluid than an amoeba. Because amoebas are slow and kind of gelatinous, but this thing is really fluid and fast. See how much it stretches out? Like a long proboscis. These guys are crazy. I know he's stretched over on the other side. Way on the other side of the debris there. That's hilarious. Sit back here and watch them. It's so much fun. Hey Catherine, how's it going? Sorry, I'm way behind on chat here. This is what is this, third, fourth stream on the same water samples from the zoo from well over a week ago, right, 2 weeks ago at this point. And it was more of just a, hey, I have samples in the fridge. I wonder if they still have any goodies in them. So here they are again, and went through several different numbered samples based on where they were collected in the zoo, and this sample number 4 always has the best stuff. It's this filter trap in a flamingo pond at the zoo where I used to work, and every time I get water samples there, it has the best stuff. Occasionally I find some new things that I haven't seen or for a long time or forever sometimes. So I just had a few things cruising around earlier. There's a long cilia called a Spirostomum. I'm pretty sure I have to look at that one up, but And this is this one called, I believe this was Amphileptus. There are a few that look like this. But just look at this, is again, it's just like the Spirostoma. This is a single-celled animal, incredibly flexible. Look at that front end, looks like a proboscis, acting very multicellular, but it's just a single cell. It can change the shape of the cell so much and so quickly. It's just a really fun specimen to watch. It's like it's having a fit, and it's constantly changing the body shape and squishing that front part of the cell out. You watch, I love paramecia, but they're pretty boring compared to these things because they don't do a whole lot as far as they're kind of stiff. They can flex a little bit, kind of smush into things a little bit, but that's the most you ever get out of them. These guys are actually changing shape and bending around things and extending the front part way out like a tentacle or proboscis. Just paramecium, it's got nothing on these guys as far as activity and just bizarre behavior. And same with that Spirostomum thingy that was cruising around earlier. Super long, very flexible, very worm-like, but again, one giant cell. It's almost like nobody told them they're single cell, they have to behave like a single cell. It's like, no, I've got abilities. I'm gonna flex, I'm gonna twist, I'm gonna bend around stuff, I'm gonna do all this stuff these other guys can't do or don't seem to learn how to do, some of these other ciliates. But these guys have some tricks. It's very interesting. So it looks like Catherine left, but thanks for dropping by. I hope you watch the rest of it to catch this. And some of my explanation here of what's happening. Pretty cool. Well, now it's embedded in there. It's not much to see anymore. So let's go find the big guy one more time. And then I'll look at the big samples I brought to look at in the other scope. Looks like the big guy has moved on, way on. I don't know where it went. Could have gone anywhere. There's that shrimp antenna again from the little shrimp that the flamingo are eating. A piece of the antenna. Stalk and all those little bristles. That's an idea of scale, basically. These are very small shrimp that they eat, and look how big that is. That's just the antenna compared to some of these other things you're looking at. Again, this is 100 power plus a little extra for digital zoom, just to get a little extra detail. So I give you an idea of the scale. Oh, nice, one of these guys again. Very cool. I haven't identified these yet either. I have a rough idea what it is, but I need to look it up. It's about the size of a paramecium, a little smaller. I find them occasionally. I've actually had recordings of them dying on a couple of the streams because they're fun to watch for a while, and then all of a sudden sometimes they'll just stop. Contractile vacuole just stops pumping, and then they start inflating with water. A good old osmosis, and then they pop. They don't have any real distinct markings other than there's a lot of stuff going on inside the cell when you get a nice close view of them. And they often have eaten a lot of this colorful stuff, so they tend to have some nice colorful food vacuoles inside of them. And they do this tumbling behavior too. You see them kind of twist on their axis, which is a very nice effect. It's almost like a little snow globe. All the stuff moving around inside. So they're very fun to watch, but they're not like really weird or any special markings on them or whatever. They're pretty much like a little cold capsule with some details inside and just kind of spinning around like that all the time. They don't have a whole lot of stuff going for them, but they are fun to watch, and they're pretty common in this, especially in these samples I've been getting from the pond. Just need to identify it. not too fast. They kind of hang around a lot. They spin, they tumble. So there you go, this little spinning action. So again, easy to find, big enough to see some nice details inside the cell, but the cell itself is not too remarkable as far as weird shape or behavior or anything like that. You can see how it's nowhere near as flexible as the last two things I showed, which are bending around all kinds of corners and flipping around and stretching out like Plastic Man or Mr. Fantastic. Just nice little things to look at. Oops, let's do a little more dark field of this guy. Some contrast. So you can see all this stuff inside the cell there with a little more contrast. Bright light, natural look to it here. This is what they look like in your typical scope view, so quite a bit darker. All the stuff inside. Oh, thanks, Henrik. Thanks for dropping by. Have fun. Come back later. I'm going to be looking at some stuff from the fridge, some produce and some other food bits and stuff under the other scope, just for fun. Oh, this guy's eating a little diatom. There's a diatom inside there. A long, skinny one. Yeah, this because I was talking about the snow globe effect with this one. This one is really a neat species because it has there's just so many particles inside. They're really obvious. It's just got a lot of stuff going on inside the cell, and as they smoosh through things or rotate and tumble and stuff, it just looks like a little snow globe on the dark field setting. But on this setting, you can really see the individual dark particles sloshing about inside. It gives you a nice sense of 3D, like stuff suspended inside a water glass, just spinning around, or a snow globe, like I said. It's just a lot of things I could just stare at this for hours, just because it's a lot of complexity in this cell. Even though again, I said the cell is fairly nondescript. They're kind of plain as far as their behavior and stuff, but you really have a lot of detail inside the cell when you get a nice clear view. Just watch them move around. Let's fix this in a bit here. tend not to move too quick. They'll take times like this where they just kind of poke around and probe and spin around, and so they're really easy to study. It's very mellow. I don't have to sit there and chase it like a video game. Very nice. Alright, let's go looking for a couple more things. Or to slide. Yeah, see the water's already coming in and starting to squash. Do the big crunch as the slide is evaporating. There's that weird structure again. There's a shrimp antenna. Oh, there's a vorticella on the shrimp antenna. That's why it pays to switch sometimes to a different view, like a bright field, because you can find guys like vorticella here very easily on a bright background. Little wine glass on a stalk. It's got the mouth parts open at the top, even though it's a single cell on a stalk. It has the cilia fanned out, kind of like a rotifer, but rotifer's multicellular. And they've got that funny little thread that they coil up for defense purposes or whatever, but he's about to ready to get squashed by a front of water here. He's not going to be very happy when that comes in. Not going to be very happy at all. Oh, there's actually thread's going to get squashed. You won't be able to recoil. This will be interesting. Now it's pinned. It can no longer recoil. I've never seen this before. What's it going to do? That water pressure is a lot of pressure as that air bubble. The air's, pushing through the water. The cohesion of the water wavefront there is incredibly strong. And now that guy's thread is no longer able to retract. The cell itself is fine. I've seen these guys, they break off the little stalk all the time. See, it can only retract that tiny bit. It can no longer retract to the base. That is a trip. I've never seen that before. And so it's fine, and it could actually detach from that stalk and free swim and then find somewhere else to regrow stalk, essentially. I've never seen them do the thing. I've seen them free floating, swimming around, and then either something got the stalk or they shed the stalk. I'm not sure how that works. They're so much fun to watch. Just sitting there filter feeding just like rotifers do. Just on a microscopic level, doing exactly what clams and mussels do and all that's just opening up and filter feeding on small bacteria and tiny little protists, mostly bacteria at this scale. Because you'll see them reject things. They're creating a called vorticella because they create a little vortex with the pumping cilia, which you can't really see at this scale on this lighting. You can't see the cilia moving so much. You just see that crown thing open up, and that's when they're feeding. And you'll see small things come in, get sucked in, and bounce off. That's like rejected by size. And these guys also do what the other things I showed earlier were doing. They have size rejection, so things will bounce off. And once they get into the little gullet area, I'll zoom in a little bit more so you can see some details of this poor guy as it's getting nowhere else to go. Here comes the water. Now he can no longer retract because it's pinned and also cut off. See, it's trying to retract back, but it can't. See the cilia flap around a little bit there, but it's trying to retract and it can't. It's probably like, what the hell? Why can't I go back there? And again, the waterfront's going to continue to advance and eventually cut off the... It's not going to kill the cell, but it will keep it from... Basically water will squash him. There's another tiny vorticella below here. Yeah, he's about ready to get smashed against this shrimp head, the shrimp antenna stalk. There's a tiny vorticella toward the bottom, you can see. Down here, it's the opacity's a little bit off. I can fix the exposure to show it a little bit better. Oh, he broke off. See, look, vorticella's free. It came off the stalk. It knows how to get off. So that's cool. I've actually never documented that before. It cuts itself off at the stalk, and now it's running to the other side next to the other tiny vorticella further down. There it is right there. The little tiny vorticella is right there. So you can see the size difference. Could be a different species. It could be just a... I don't think these guys grow that much, so it's either a slightly younger one or a different species of vorticella. But yeah, so this guy, now it's upside down like a little hat. So the little mouth parts down here, to separate it when the water started to squash it, it's separated from its own stalk and went forward. And see if it stayed behind, it'd be squashed right now by that waterfront, hitting the huge shrimp antenna. It's just chilling out there. But it's funny because it attached to that, it went forward and the mouth part, kind of little cavity is still open. Like it's trying to feed. But it's kind of stuck against that debris. It's not moving too much, but it's moving a little bit. I don't know what it's going to do now. But see, the wavefront is still coming, so it doesn't matter. It's still going to get squashed if it doesn't get the hell out of there. This little one down below is still attached. Here comes the wavefront. What's it going to do? Is it going to move again? Got some drama here. Need the suspenseful music. I should have my keyboard hooked up. Took it slow a little bit, but the water is still advancing. Oh, there's free floating. There it goes. Oh, now it's attached to the antenna. But it's did the wrong move. It actually went further up, which is back toward where the collapse is going to happen in the wavefront. So, bad move, buddy. You should go the other way. You got your freedom, and now you're going to get squashed. And there it goes. He's about to get squashed. Now there's nowhere to go. There's a pocket of debris at the bottom. It's going to keep the water from going all the way in flat against the antenna. So it's actually kind of funny. It's like a barrier now. But now he's stuck in that cavity. He can't get out. And now the little vorticella at the bottom is going to get touched by the waterfront. The little wavefront coming in. Didn't notice it yet because it didn't touch it. Further and further in. Now the waterfront is touching the small vorticella. It's still on a stalk down below, but it doesn't seem to bother it. Oh, now the waterfront's coming from the other side. Oh boy, it's not looking good. debris slowed down the left side advance, but the right side is not coming in. And now the little guy's retracting, but oblivious to the fact that pretty soon there's nowhere to retract to. The other one's just pinned at the top there. Not doing anything. And it could be this way for another several minutes. I'm not going to sit here and watch the demise of the poor Vorticelli. You get the concept here. But let's go further down and see who else got stuck. I've got Amphileptus. Whoa, look how stretched that thing out. Whoa, I've never seen it stretch out so much. Look at that. Holy moly. That's just like Plastic Man or Mr. Fantastic. That thing is stretched out like 5 times the length of the cell. That is crazy. Let's go some diatoms at the top there. So again, the debris pockets, another good reason to hang on the debris pockets from these guys' point of view, although they don't know it, is when the water starts coming in. See, it's all on all sides here. And it delays the crunch effect of the water coming in and squashing these guys. So they don't have infinite time, but they have quite a bit more time than they normally would have with the samples I've collected where the water just comes in and squashes everything down to a single air bubble with some debris, and then the thing is like being shoved in the trunk. There just is no way to move anywhere. So it's going to keep getting squashed. Vorticello's still there, just pinned, nowhere to go. Little guy's still there, oblivious to what's happening. There's some algae there. It looks like everywhere else, there's some pockets over here. Those didn't dry up yet. Got some flagellates poking around here. I don't see the big Spirostomum guy here. Wonder where it went. It may still be free. There it is. See, it doesn't have a care in the world. It's like, doing my thing. What waterfront? I don't see any waterfront. There's nothing coming to squash me. It's just here doing its thing. Business as usual. So you can see it twisting. It's very flexible. Back end is very flat, kind of twists around like a ribbon. Now it's going backward. Getting very wormy behavior for a single-celled animal. Cool. Alright, I'll give these guys a break. Let's go on to some big stuff. Switch scopes and do some big stuff. Some stuff from the fridge. in the kitchen. Some macro things. Switch cameras here. So there's the zoom out here. It's pretty much maximum zoom on this guy. Maximum zoom out. So just to give you an idea what I was looking at before, here's the chocolate stuff. Nutella spread. So see, as a mass, it's this brown, slimy stuff. When you thin it out a bit, it still looks brown, but it's much lighter. And you saw in the compound scope what it looked like, where it was very little color at all. It's mostly, you know, some white bits with some little bits of color, chocolate color flakes mixed in. But then if you, let's try doing the backlighting on it here. Turn this down. And let's adjust this exposure here a bit. Oh great, so it's stuck again. Come on, software, there we go. Let's adjust the contrast here, brightness here. So, shining light through it. This is similar to the effect you get in the compound scope, but then I can turn that off and just do the top down, or a combination, and look at specimens in a couple different ways at once. Now I can zoom in to the mass, get a little more light, a little more exposure to the camera, and these are the, sometimes you have to actually readjust the whole unit. Some more vertical action there, and then I have more room to focus it. If I go too far out, then I can't focus at all, so I have to bring it back down. There we go. So there's the slimy chocolatey goodness that is Nutella. See, it's all kind of bumpy because it's essentially you know, oil, emulsified oil, sugar, chocolate powder, hazelnut flavor, stuff like, it's pretty basic stuff, but you can see it's just this fatty, it's a very fatty spread. It has those kind of fat globules making this kind of transparent lumps with some brown color when it gets concentrated enough. Just trying to change the backlight a little bit. And I have a nice mixture of both. You can see the texture as well as the color when I do the backlight. So that was Nutella. This was the calcium carbonate I smashed earlier. Here's a piece of the Tums and then the powder itself. There's the big old piece of the tablet itself. Very crystalline, chunky. Could use a probe here to poke around a little bit with it. So nice clean edges when they compress the thing down to a tablet on the edge, the round edge. You can see where I broke it. It's very fragmented. Let's pop it on an edge. And you can see. Come on, get back here. You can see. It just looks like a mineral with some color veins going through it there. Berry Tums. So it's pink with some nice white chunky calcium carbonate crystals and some other finer bits mixed in. The big things are actually probably sugar crystals, I'm guessing, because there is a fair amount of sugar in Tums. And then the finer stuff is probably the calcium carbonate. And then your flavoring. It's kind of those little veins of flavor mixed with the pink pigment. You focus on the top and you get this kind of rough, like a little stone thing. And if I move over to the area where it's all powdery, this is when you smash it down to calcium carbonate and other bits. Again, I can backlight it for effect. But this is kind of fun like this, just the powdery bits with some bigger white chunks there. So that's what Tums looks like. And around, what is this? Probably, yeah, it's not that much, maybe like 30 power or something like that. So that's fantastic. So if you have a lot of chocolate before you go to bed, you might need a Tums. That's why those two go well together. This chocolate, especially if it's stronger chocolate, it's pretty acidic. So yeah, that's that. Then we got, what else we got? Here's a piece of apple with the, where's that little tray I had here? Nice tray for this part of it, so I don't get slimy bits on my scope. Seem to have misplaced my little tray. Where'd it go? Not there, not there. Hold on while I track down my tray. I only had a couple of them. Give me a second. I might have moved over by the sink to clean it up a little bit. I'll be right back. That's weird. Just getting all kinds of stuff prepped beforehand. And I had a tray just for this specimen stuff. Oh, here it is. It was under some other stuff. So, this little part of a petri dish is the plastic petri dish often used to grow thingies in the lab, like agar gel from seaweed. As a bland matrix that you can put food in and antibiotics in, all kinds of other stuff to play with bacteria and see how they react. So here's a piece of an apple. Zoom way out here. And when I eat the apple, I just do a simple coring of it. I just cut the base part out of it in the middle so I don't waste too much of the tissue. And here's the section where the seeds are located. And they always have this white material around it that I wanted to check out under the scope. It's kind of firm, fluffy, firm white material. Oops. Stiffer than I thought it would be. There's one of the seeds, which, as you all know, has a little bit of, probably not as much as like a cherry pit or something like that, but has a little bit of cyanide in it and some other stuff. apples have a lot of what they call malic acid in it, named after the genus Malus, which is the apples are part of. Well, that seed's really in there. I guess I'll let this thing dry out when I cut it earlier today. It's really hard to get that seed out of there. There you go. There's the seed. Probably too hard to open it with the probe. Usually that's pretty easy. So I can pop this thing open with the forceps and the probe. So inside the seed, you call it the endosperm, which is basically just it's the part you actually eat when you eat like a wheat grain. It's primarily just the matrix holding the little seedling inside, the micro seedling. It's called endosperm. It's just food for the seedling to grow. But yeah, here's this weird fluffy but firm things. There's a lot of apples have that. Not all the time. Sometimes it's just a little compartment with a thing in it, but I've noticed a lot of these lately have this kind of woolly but firm stuff in there. It's kind of interesting. And then we have what else we got here? Piece of lettuce. That's romaine lettuce. That's kind of going off, so I don't want to eat it. I thought instead of just wasting it, might as well have a little look at it first, just to see any interesting stuff on the micro scale. There's my probe for reference there. Let's flip it over. Starting to wilt and change color, which is always lovely. Let's do some backlighting here. Turn the front light off. You can see the venation in the leaves there. Lettuce leaf venation. The stem is way too thick for the transmission of light, so it's dark. If I turn it way up, then you can see that part's too overexposed. This part is not. It's much darker. So that's the soft bits people like. Then further down the stem, it's pretty thick. There's some lovely. When I bought it, there was clearly some insect activity on this, which started to spoil the romaine. So I tossed it, but I wanted to see what this looked like a little closer to see if you can see like little holes from like insect jaws. Perforated areas, and then it started to spoil, so you're getting bacteria and especially fungi here starting to grow a little network of rotting bits to digest it for recycling. So not that interesting, but I thought I'd take a look at it anyway. And here's a little piece of red bell pepper. Quite colorful, shiny. It's like one of those fake fingernails, kind of. And the skin has already started to curl because it's drying up, but the middle is still kind of wet there. There's a little bit of tissue still on the thing. Flip it on the other side, flatten it out. See it's very, very shiny, which makes bell peppers look so nice. I could try to do some backlighting here, see what we got going on in the backlighting. See the different colors going through the skin based on some tissue still on the back end. Some areas not so much, where it's very light colored. very colorful though. Oh, and then I had this lovely spoiled carrot. Sometimes you buy carrots in bulk. It's a very bad idea. You're saving money, but you can't inspect the whole bag. And then sometimes the bag goes bad really quickly. Like, look at this. This is awful. This bag of carrots immediately had all this fungus and rot going on here. Look at that. So it might have like some little divots in it from the picking process, because they just use, you know, this kind of large things of carrots. I think are just machine picked, so the machines sometimes do some damage. But you get this, you know, stuff starting to. It's wide open for stuff to get in here. You know, you can cut some of this stuff away if it's not too bad, and then eat the carrot. But this is so much infiltration here of the tissue that, you know, you can't, as I showed in other streams with the fungi, the hyphae, the little threads that basically form the basis of the fungal body, and eventually becomes like a hairy patch, and then grows like roots into stuff and forms a giant mycelium, and then eventually it will grow fruiting bodies, which are like mushrooms. And look how much stuff I have to pick away, like your dentist picking a bad tooth, just to get to where the carrot would have been somewhat edible. So it's a lot of cleanup just to make this carrot somewhat edible, and even then you're not sure because the mycelium go way down into this thing. You're not seeing them so well here because they're very small, the hyphae. But once you have this kind of damage and the fungus is going nuts, it's like, yeah, it's a good time to throw it away. But hey, I got to use it for a stream first before destroying it, because why not? So I get all this nasty fungal tissue growing over the cut, basically. The carrot wasn't able to close its cut properly. It's all kinds of moisture here, and the fungal hyphae just go nuts. They go, wee! Look at all this. We need moisture and we need nutrients. So carrots are very sweet. So of course I had a good old time making a big mess here. So yeah, in this case you say, oh, I could just cut those parts off. Nah, I wouldn't do it, especially when there's this much rot on it. Just toss it. Carrots aren't that expensive, and just don't do it. Here's the tip of it, which is really bad. Ugh, look at that. Awful. See, it's a bit woolly there. That's mycelium. Form this fungal mat. It perfectly covers up the area, so it grabs all the moisture and stores it. Makes a nice seal. On the outside will eventually grow spores and get all powdery and continue to spread into other areas on the wind or from touch. So yeah, nasty carrot. You know, this part at the bottom's not so bad. I got it away from the actual, where I broke it off the carrot. You can see the centerpiece there. Well, that's normal carrot pretty much there. It looks okay. But all this crap around it, it's like, yeah, no thanks. You have to cut so much of it out, and you can't be sure you get most of the nasty stuff. You just don't want to eat all that. It's best to throw it away. So you have that, and then you have the red pepper itself. I had that little piece before, but here's the actual stem I cut off the part so I can make a salad with it. And all the seeds attached inside. All the seeds still attached. Nice little rows. Looks like little scalloped potatoes. So if I zoom way out, it's about as far as I can get on this guy. It's pretty close. Change the lighting a little bit, and you can get a nice look at the seed structure of the inside your peppers. Very loosely attached. They're easy to break off if you hit them hard enough. But they stay here as long as they need to stay here. And then you have some tissue here, if I rotate it around just right. And then back onto the outside of the pepper is nice and shiny. The skin is very tough. In this case it's getting soft because it was sitting out for a while, so you can dimple it really easily. It's still pretty tough. It takes quite a bit to actually puncture it. You can just make some imperfections on it, but pretty tough stuff. There's the stem sticking out there. It's already starting to get some nasty stuff growing on it. And if I turn it upside down, so you can see the bottom of the seeds. Seeds are really in here. This kind of woolly matrix of kind of tear through it a little bit there. Down to the pepper underneath. See, it's not that thick. It almost looks like surgery. It's just kind of nasty. Yeah, see the seeds pop off pretty easily. Just brushing them. It's gonna break right off. I can zoom in on it a little bit. You guys get a better idea of the. They're fairly smooth. There's not a lot of detail on the seeds. So yeah, there's the pepper. Red bell pepper. I also have this other sample from a pine tree I collected a while back. I forgot to put this on the stream, I think. A little bit dry now, but get the idea. It's a lot more light. So at the base, you can see the little the stems grow, these little curly bits. And you get further up, it can actually get to where the needles start to come out in little bundles. See the bundles coming out here. There's like this little papery bit on the bundles. Little papery thing that kind of keeps the bundles together. So this is part of how you identify pines, not just by their overall size, color, cone. It's also the number of needles in a bundle. They're also often called a fascicle. So when you pull one of these bundles off, this particular pine has 2 needles per bundle. Down to the bottom of the bundle there, you can see that now it spreads out to 2 needles. Try backlight, see what that does. Get the better shape of the needles there, the backlight. So that's pine. I don't have a cone for this one, but it's kind of getting dry. It's not as green as it was before. But these preserve quite well. I mean, as you see with Christmas wreaths getting old and stuff, with fir trees and other kinds of conifers, they're obviously more fragile and will drop needles easily and also burn very easily when they're dry. As long as you store them carefully and don't bang them up, they actually last a long time as they were growing, just as a kind of a dried specimen. So, kind of nice if you're interested in dried plants. I think that's pretty much it. I went through all the samples again, got some good stuff. Went through a couple random kitchen and food items. Can't think of anything else other cool things right now. So thanks for all of you who participated and that joined in the chat and all kinds of stuff. I'm gonna have to go back to read all the chat. I got so busy, it was flying by at one point and I couldn't read it all. So thanks for participating and all that. Always happy when people show up on the streams, and of course anyone comes back and watches the recorded stream, makes comments, whatever, everything, ask questions, do whatever you feel there. And I always like the participation, like I said, and thank you for joining. And let me know what you think of the I kind of messed up the layout there that time, didn't I? It was fine for a while, and now is it still? Yeah. Somewhere during the stream I actually changed the magnification of this guy, but and I got the two cameras going now, so I got the microscope and then the live camera for this table view. So hopefully you guys like that. It makes it a little more dynamic and you kind of see what I'm doing while I'm making noise going from scope to scope. I'm going to set up a little better in the future so you can actually see things better with the camera on the table, as well as you know, switching cameras between the scopes. But yeah, that's about it. So again, thanks for everybody joining, and hope the audio levels are good and all that usual stuff. And stay curious, go out there and do some nature exploration. Get a hand lens out, get some binoculars out, take a look at some birds and some plants, and you'd be surprised what you can see even with the literally simple cheap hand lens, like a magnifying glass, or even ones you can stack to get like 5, 10, 15x in your hand. And you can even find, you know, get a little sample, a water sample from a pond, put in a little petri dish or some little glass bowl, and you know, shallow enough that you can get close to it, and put the hand lens on there, and you'd be surprised what you can see. It's just like a tiny portable microscope for looking at certain kinds of things in the water, even algae and copepods and all kinds of interesting things to look at. You don't need a fancy microscope to see some of this stuff, but yeah, I'm babbling at this point. But thanks again for everybody showing up, and appreciate it, and have a good evening, good morning, good afternoon, wherever you happen to be. Thank you.